Thanks a lot. I will talk to you about today how data analysis can help in making sense of what we measure when we measure brain activity. In particular, when we measure brain activity with the functional magnetic resonance imaging. So what is functional magnetic resonance imaging? Is a very powerful tool which has rocketed in the last years, as you can see from the red curves. And it is indeed so popular because it's non-invasive, it has a great spatial resolution, and to some it's easy to interpret, but indeed it's also very challenging and easy to misinterpret. Indeed, if we want to measure brain activity, brain is made of neurons. If we want to measure activity in all the brain in a living human being, we should either open up the box or find an indirect way, which is indeed fMRI, which measures the level of oxygen. Where does it measure it? It measures it like in the equivalent of pixels in the brain. So volumetric pixels, say it's voxels, and we have several thousand to millions of them in the brain. So this already gives you the idea of what is big data in neuroscience. So in the ideal case, the brain will tell us exactly how it works and how it responds to stimuli. On the other hand, of course, it's not like that. And what you observe is still an idealized case that you see in newspapers when they talk about brain areas that light up when some stimulus is presented, and they show the figures like the blobs. And indeed, you can imagine like really brain areas that starts to pump up like crazy. On the other hand, in reality, this is what happens. So activated and non-activated areas are indeed look like the same. That's why we need statistics, and we need good statistics. Otherwise, what we will observe in our blobs, there will not be a sign of real brain activity, but it will be just a sign of poor statistics. So this is the first application of statistics and big data. Then it comes to connectivity, because it's great to know what happens in each area of the brain, but we actually need to know how the things merge in order to make sense, to give rise to function in the brain. So indeed, brain function emerges from the correlated activity of areas which are functionally segregated and specialized. So there is area for vision, for memory, for olfactory area, but they all together go to build memories and sensory function. So connectivity also has picked up much faster than activation in the last year and allows to look at brain activity in terms of a graph. And in this case is analytically treatable, as we have seen before by excellent talks. So if we want to infer the network from temporary correlated data, we can look at simple correlation or other fancier measurements, but these measurements are bidirectional. On the other hand, what we need is directionality, because if two regions are correlated, it can be that the flow is from A to B or from B to A, or both A and B are under the effect of a third region C, and this is very relevant for brain function. So in order to solve this problem, two methods have been developed. Transfer entropy, which is based on probability, is a model-free method, and Grange causality developed originally in econometrics, which is based on autoregressive models. But the problem of these two methods is that when it comes to conditioning, so to separate mediated from directed influences, encounter problems when we have a huge number of variables and short time series in the framework of autoregressive models. So that's why we have to use information theory and rank the variables according to their informational content. So in the brain, it turns out that we can use only 10 or 20 variables, it's enough. And these variables are also consistently distributed in the brain. But when it goes to huge data sets, so for each voxel, then we have to use an extra structure. So the Louvain algorithm, another made in Belgium, extremely powerful tool. So once we have found the network structure at the voxel level of the brain, we can constrain the distribution of maximal informative variables by the modules of brain architecture. So in this way, we were able, in our lab a couple of years ago, for the first time, to construct the network of directed information flow at millimeter scale. So here you see the modules of this network, and on the right you see where these modules are located in the brain, giving a lot of insight on brain function in terms of networks. So the take-home message for you is that powerful brain imaging as fMRI can become useless or misleading without appropriate analysis. Structures must be coupled with the local and global function. And in this case, information theory and graph theory will do the trick. So thanks a lot.